Hi, everyone. Good morning. We're going to get started. Today, I'm excited to welcome everyone to a very special Grand Rounds, the third annual Dr. Barbara Murphy Memorial Lectureship. To begin, I'd like to introduce Dr. Monica Kraft, the System Chair for the Department of Medicine, to say a few words.
Thank you all for joining us today, both here and on Zoom, and for the third annual Barbara T. Murphy Memorial Lectureship. You know, although I never had the privilege of meeting Dr. Murphy, all accounts I've heard really paint a picture of an extraordinary person, an inspiring leader, a pioneering scientist, and a kind, compassionate human being. It's really an honor to celebrate her today and the impact that she has made on Mount Sinai and her immense contributions to nephrology and medical science. Her work and vision have left a lasting impact on our patients, colleagues, and fellow researchers. And the Division of Nephrology at Mount Sinai, which is an incredibly strong division, I'm very proud of them, continues to advance her mission of building a healthier future, engaging in groundbreaking kidney research that saves countless lives, which is why making this important lectureship named after Dr. Murphy a reflection of the indelible mark she has made here at Mount Sinai and beyond. I also want to acknowledge Dr. Peter Fogarty, Dr. Murphy's husband, who is on Zoom with us today, and thank you, Dr. Fogarty, for being with us. I'm also delighted to welcome Dr. Norm Rosenblum, which we'll hear more about from CJ He, Dr. He, a renowned scientist and leader in pediatric nephrology in Canada. Through groundbreaking research in genetic and molecular mechanisms that regulate kidney development, he's really revolutionized the field. Dr. Rosenblum's innovative use of genetic mouse models and stem cell-derived human kidney organoids have propelled our understanding of renal development and disorders. And beyond his scientific achievement, he's also a cherished friend, whom I had the honor of meeting through our mutual interest in translational medicine through the Eureka program, which I think some of you know about. Norm, thank you for being with us today and sharing your extensive knowledge, the work that you're doing at the Hospital for Sick Children at the University of Toronto and the Canadian Institutes of Research. It's a privilege for you to be with us, and you're the perfect lecturer for the Barbara T. Murphy Memorial Lectureship. So with that, I'd like to have Dr. He come up and do a more formal introduction of Dr. Rosenblum. Thank you.
Morning. First, I'd like to thank Dr. Kraft and the Department of Medicine to support this event. This is a really very special occasion for us, especially for those who have been working with Barbara for many years, for the faculty in the Division of Nephrology. Since we have a lot of new resident fellows and maybe some faculties, so I'd like to give you a little bit brief introduction on Barbara's, what Barbara she means to all of us. So Barbara was born on October 15, 1964 in Dublin, Ireland. Barbara attended medical school at the Royal College of Surgeons in Ireland and completed internship residency clinical fellowship, clinical nephrology training at the Bowman Hospital in Dublin. Then Barbara decided to pursue a further training in the United States, where she completed a transplant immunology fellowship in the laboratory of immunogenetic and transplantation at the Brigham Women under mentorship of Mohammed Sayed, who is a giant in the kidney immunology. Barbara was recruited to be the director of the transplant nephrology program at Mount Sinai in 1997, where she built one of the most robust transplant nephrology programs in the country. In 2004, when she was 40, Barbara was appointed chief of division nephrology at Mount Sinai, and during the next 8 years, under her leadership, the division experienced a rapid expansion into clinical care, training program, and NIH funding. As a faculty in the division, we all benefit from her leadership and mentorship. Barbara was promoted to chair of the Department of Medicine in 2012. Barbara was instrumental in integrating the clinical and the training program for several large hospitals and became the first system chair of medicine for the Mount Sinai Health System. As an internationally renowned academician, Barbara served as the president of the American Society of Transplantation and was the president-elect of American Society of Nephrology in 2022, just before she passed away. Barbara received numerous honorary awards and doctorate degrees from academic institutions and a variety of societies, including the inaugural American Society of Nephrology Trailblazer Award for her extraordinary contribution to patient care, research, and education. So as a scientist, Barbara was an international leader in the field of genomic medicine. Her groundbreaking research used high-throughput genomic technology to understand the immunomechanisms that lead to allograft injury and loss. And under her leadership, 2 companies were also formed at Mount Sinai to develop AI-based diagnostic tool to improve the CKD education, CKD detection, management, and also the treatment. Barbara was also a great mentor. Numerous trainees from her laboratory went on to become funded investigators, assumed the leadership position around the world. Moreover, during her time as division chief and chair of medicine, Barbara was really unwavering in her support of the 100 faculty fellows and residents, including myself, who actually benefited from her leadership and guidance. Barbara's brilliant professional achievements were unremarkable. However, for those who had the great fortune to know her, it was not those achievements that made Barbara so special and beloved. She was a nurturing mentor who prioritized the personal happiness and well-being of the trainee and the faculty in addition to their professional goals. Barbara is a genuinely humble person and a loyal friend with a lot of humor. So now, in her memory, it's my distinct pleasure to introduce the 3rd annual Barbara Murphy Lecture, Dr. Norman Rosenblum. So Dr. Rosenblum is a pediatric nephrologist and senior scientist at the Developmental and Stem Cell Biology Research Institute at the Hospital for Sick Children. He's also a professor of pediatric physiology and laboratory medicine and pathophysiology at the University of Toronto, and the scientific director at the Institute of Nutrition, Metabolism, and Diabetes of Canadian Institute of Health Research. The major focus of Dr. Rosenblum's research is genetic and molecular mechanisms that control normal and disrupt the renal development by using genetic mouse model and the stem cell-derived human kidney organoid. His research team has generated several important models for human kidney urinary tract malinformation and has published over 150 peer-reviewed original manuscripts and book chapters. Dr. Rosenblum has been well-funded by Canadian Institute of Health Research. He has a multiple leadership position and served as a member in many committees. As the scientific director of the Canadian Institute of Health Research, he has led the development of the largest Canadian federal investment in diabetes research. Dr. Rosenblum is also a great clinician and educator. He has trained many mentees. He served as Associate Dean for Physician Scientist Training in the University of Toronto and a member of the Steering Committee of the NIH-funded Pediatric Scientist Development Program. Dr. Rosenblum really exemplified the very best of academic nephrologists and is a perfect candidate for this lecture. Now, please join me to welcome Dr. Rosenblum to give us a lecture on Nephron, Endowment, and the crisis in chronic kidney disease. Thank you.
Well, good morning, everybody, and thank you, Monica, Dr. Kraft, my good friend Monica and John. Thank you very much. I very much appreciate this invitation. I mean, really, it's an extraordinary honor to be here. Unfortunately for me, I never had the pleasure of meeting Dr. Murphy, but I became aware of her and began to read about her during her lifetime and afterwards, and it is truly extraordinary to read about the breadth of the person that was encompassed by her, and I really thank you for this great honor to be here. In reading about her, I realized she and I had a lot in common, even though she's great and I'm, okay, but there's a lot about her that I would emulate, and as I read about her, I realized, well, she was inspired to nephrology because of patients. And I had a similar experience because of what patients were experiencing and the gaps in our knowledge. and that inspired me to want to be a nephrologist. She trained at the Brigham and Women's Hospital in Boston, which was right across the street from the Children's Hospital, where I trained, and in fact, I did a fair bit of service in neonatology at the Brigham and Women's, and collaborated with people in nephrology at the Brigham and Women's. She initially planned to be a clinician, but transitioned to be a clinician scientist. That's exactly my story. I was inspired by how much ignorance existed in what we knew how to do, and I said I just couldn't stand it anymore and was going to try to do something about it. She was a trailblazer. Well, I'd like to be a trailblazer. She was definitely a trailblazer. She harnessed genetics, and I've lived through this wave of developmental genetics and genomics. That's really the story of my entire career, starting in the late 80s when the world was completely turned upside down by these disciplines. And I've been riding this wave for about 35 years. I think many of us have to reach this point in time. And finally, she was interested in nephron loss and how to replace nephron function. And I think certainly I'm interested in that and am very motivated by the challenges that we have. So I really identify with her career. And as I said, it's a tremendous honor to be here with you. Here are the things that I'd like to talk to you about for the next 3 hours. First, we'll just quickly summarize the challenge of chronic kidney disease and understand its place in human disease at present. Then we'll focus on nephron number and the challenge that, what I'll say, lies beneath the water's edge, the sort of invisible challenge, if it were. Then I want to acquaint you with some general experimental approaches that my lab uses, and I think a lot of people use in different disciplines to try to address the control of nephron number. And then we're going to sort of end up with what I'll call what's beneath the water's edge, which is the hundreds of thousands of individuals who are being born into our societies who very likely have a deficit of nephron number and what the implications of that deficit could be. And then we'll finally harness Dr. Murphy to focus on about 4 major gaps and opportunities that all of you who are younger than me, which is basically everybody, can harness for the rest of your lives, because these are big problems that our societies face in a general sense, but also in medicine. So let's begin. Chronic kidney disease is a common ailment in the population. Approximately 1 in 10 individuals is suffering from chronic kidney disease. I know that you in medicine, as opposed to pediatrics, you'll be dominated by diseases like diabetes mellitus, which is such a common determinant. But these are big features where it's very common in individuals with diabetes mellitus. It has a differential effect across populations, for example, racial minorities, more prevalent in women, more prevalent in the elderly, certainly prevalent in individuals with hypertension. And the problem that we face is that it's just getting worse. And it's rising, predicted to rise from 1 in 10 to about the 5th major cause of morbidity in Western societies as time moves forward. And here's some data from all over the world. And you can see that it is variable in terms of the proportion of national expenditures, which is a bit of a crude way of indexing how important this problem is. But look how it eats up the economic prowess of various societies. You can see some societies are just dominated up to the point of like 10 to 15% of GDP by the occurrence of chronic kidney disease. And these bars are meant to show the level of spending in 2022 and the predicted levels in 2027. And you can see in some of these societies it's just getting worse. And maybe some of these places are not the ones that you automatically think about, but it's important to think, like Mexico, which is high altitude. Why is it that, for example, the prevalence of CKD is so high in a society like that, or in Thailand, or in India, et cetera? It's a big global problem. And of course, as I said, one of the major drivers in medicine is diabetes mellitus, and that's getting worse too. So global prevalence of 4.7% in 1980, 8.5% in 2014, and every meeting I go to as scientific director of what you call NIDDK, we call it INMD in Canada, international meetings, policymakers, everybody's got the same story. Rising prevalence of diabetes, eating up people's lives, eating up economies, predicted to get worse, notwithstanding new drugs that are exciting and are really important in terms of their effects, we have not yet hit the prevention curve to flatten it down. So these are major problems that we face. And I might emphasize maternal diabetes. 1 in 14 women have type 1 diabetes, type 2 diabetes, or gestational diabetes, and there's an increased risk of childhood chronic kidney disease in the progeny. And now stay tuned on the progeny part, because we're going to stay with this theme. So as a pediatric nephrologist, staying on the progeny theme, my world is here. This is my world of children who have congenital anomalies of the kidney and urinary tract. Your world is over here as internists, where everything else is important. But my argument to you is that that's what you're facing today and what you see. But as we'll see later, I think you're going to see something else quite different as the years go by. Not that these causes won't be important necessarily, but that new things will erupt. And this is the argument I'm making. Here's the iceberg above the ocean. Well, clinically manifest disorders like diabetes and other disorders, but down here are the emerging effects of low nephron number, which expresses itself slowly but surely in certain individuals as the life course ensues. And I was talking with John just before this talk, saying the time has come, and this is a great example of where we need to be thinking life course, not pediatrics, medicine, geriatrics, but we need to think about complete flow through, because now we have the ability to see things in this way, and we have to be thinking in this way. What happens to a woman before she's pregnant, when she's pregnant, what happens in utero, what happens after birth, flows all the way through to later life. And we need to adopt this way of thinking. Well, the problem of human nephron endowment, or the challenge of it, became apparent to us when some pathologic data became obvious, showing us that actually, in contrast to what I was taught when I went to medical school in the 1800s, and that was that everybody has 1 million nephrons, and that's the way it is. Well, it turns out that's not true at all. It turns out there's about a 10-fold variance in the number of nephrons that people are endowed with, which is an in utero phenomena, complete by 36 weeks, maybe 34 weeks, depending on the individual. And so this is the variance that occurs in individuals. And several studies showed this in autopsy studies, and moreover, people really don't know why this is. Why would it be that there's such a variance in the number of nephrons that are formed in utero? Well, some people think it would be genetics, and there's some scholarly work that relate that to, for example, alleles in PAX2 or in the RET gene, but we have very little information on this. There's some information that suggests that epigenetics may be important. I'll come back to that. And we're very concerned about environmental effects, but we have very little understanding of environmental exposure and what happens with respect to nephrogenesis. So there's issues of nutrition. Certainly in animals, if you sneeze on a rat and give them a low-calorie diet, or they'll have low nephrons. But how this relates to human beings is a bit of a different question. Maternal smoking is important. Alcohol ingestion appears to be important. Glucocorticoids, which are commonly given in pre-birth situations, may be important. Other drugs, maternal health in general, and placental function. But these are big, broad categories, sometimes specific. The data are really quite weak, but they're indicators of things that may be going on. But the paper by Keller et al. In the New England Journal in 2003, which associated low nephron number in patients with hypertension, was really, I think, the wake-up call that made people start thinking about this. And then there's been, I mean, literally hundreds of animals that have been demonstrated that if you do something to them, they'll develop low nephrons in the pups. But the human data really started here, and we're left still pursuing what the causation may be. The implications of this, particularly thinking about adults, really came from Barry Brenner, who's one of the great nephrologists of our time. And it was really the so-called Brenner hypothesis that made people start to connect the dots and think about what low nephron endowment means. And what Brenner advanced was that through issues of sodium and fluid retention, extracellular volume increase, arterial pressure increase, and increased filtration through the remaining, so the glomeruli that do exist, that glomerular hypertrophy would occur in glomerular injury, and that over time, as new issues took place, like aging, loss of further nephrons, which does occur with aging, diabetes, hypertension, you name it, glomerular nephritides, you would end up with glomerular injury such that you'd have a serious level of chronic kidney disease. So that was really the Brenner hypothesis that connects low nephron number to lifelong morbidity. So let's look at some of the ways that people like myself and others are looking at nephron number. So I'll tell you, I'm going to tell you two short stories that really connect this. So I like to show this, particularly for the residents and fellows and research fellows, because when I started my lab, which was in 1993, my mentor, and I still have one, my mentor, who I took to lunch so I could tell him what I was doing and see if he thought that was great or he thought it was the most boring thing on the planet, he said to me, well, Norm, you know, you're not here to just do a bunch of experiments. What's the program of your research? What's your vision for what you're trying to accomplish? So I took out a napkin. We were at a cheap restaurant because I was paying, and we took out a paper napkin, and this is what I wrote down. This is exactly what I wrote down. I said, well, here's the thing. The kidney is the most interesting organ in biology. Sorry about that. But no kidneys, no lungs. I mean, let's face it. And so the kidney is the most interesting organ. All the developmental paradigms are shared by many organs, and the kidney is a great instructor about how this happens. So I just want to know about how the kidney forms. We don't really understand very much in 1993 about that. And then we have all kinds of genetics that are showing us that certain genes are mutated in individuals who have congenital anomalies, which is the most common cause of kidney failure in children, which is my occupation. And we don't know what these genes do. We just know that they're mutated, or variant, as we would say today. So I want to use the tools of developmental genetics to figure this out so that I can say something about diagnostics, maybe therapeutics even would be exciting, about nephron number and how the collecting system forms and functions. And that's what I wrote down. And interestingly, in every grant I've written for the last 31 years, in a steady flow of funding, this is the way it begins. Same story. Everybody knows, oh, here he comes again. Same story. Different genes, different processes, same story. So think about that when you're thinking about your future and your story. So, and I've already mentioned that, you know, we have proposed effectors of nephron number, but we're not really sure what's going on. So today we'll just focus on two stories. I'll tell you one story about a pathway that we've uncovered that controls nephron number in mice, and then some using human organoids, tissues from stem cells, exploring epigenetic regulation. And just to capture your knowledge of renal development, which I'm sure is right at the top of your barrel, I'll just review a couple of concepts so that you can understand what I'm saying. The kidney is formed from an interaction between a epithelial duct called the uretary bud, which a mass of mesenchymal cells, which really contains two populations. One is the stromal population that express FOXD1, a transcription factor, and another set of cells which express 6.2, another transcription factor, and they are nephrogenic progenitors. And when the uretary bud interacts with this tissue, then the nephrogenic progenitors start forming the nascent nephrons, and the uretary bud starts branching. The branches become the collecting system, collecting ducts, calyces, pelvis, and the nephron progenitors will form the nephrons. So, and the stromal cells start talking to both. So they're important, adjunctive cells, instructive, and they also differentiate into 5 different types of cells themselves. So that's sort of the basic story. That sums up about 30 years of work. And so here they are, lineage-wise. Here's the starting cell in the intermediate mesoderm, OSR1, 6.2, FOXD1. Here's the collecting ducts. They're interacting. And with that, you get a whole program. And so we know stuff about this program. There have been people working on this program for a long time. And there are many genes, as you can see them, I'm not going to go through them, that regulate these interactions. And one of the major signaling pathways is the Wnt signaling pathway, wingless, but there are others, and they're all very important, and we could talk about that at another time. My work has really tried to harness the fact that we work on genes that are mutated in syndromes, in human syndromes, which feature kidney disease as well as other non-renal problems. And the reason we pick syndromes is because you can identify a gene, and in this case, it's going to be a transcription factor called GLE3, which functions in the hedgehog signaling pathway, and it causes human disease. And so there is no argument about whether this gene is interesting. Because if you write your grant about Prox 1438, and the panel says, well, why are you so interested in Prox 1438? And you say, wow, it's very interesting transcription factor. It's got a loop, it's got a this and a that. But I don't really know if it's connected to human disease. The points are falling immediately. But when it's a human disease gene, you've got a starting place. So this is a gene that's associated in mice with this kind of output, which is not a good-looking kidney. And it turns out it's a very interesting gene because it's a transcription factor in its full-length form. It's an activator of transcription, but when it's processed naturally inside cells, it forms a repressor, a shorter repressor. And this occurs through proteasomal degradation, which is a natural, highly regulated process. And it turns out that the way this gene works, it works in a pathway. And it works in a pathway such that a cell surface protein called patch1 regulates another protein, smoothen, which actually promotes the pathway. And when the pathway is promoted, GLE3 activator goes into the cell and activates transcription. But when its ligand is absent, then the activator is broken down into the repressor, and it represses transcription. So here's when the ligand is present, and it blocks this repressor, so this active guy is smoothened, allows the activators to go into the nucleus. This is a very complicated pathway, very important to cancer, very important to kidney disease, and it's highly regulated, ligand dependent, but it's got activators and repressors, making sure that all the levels are just so, or else cells really go haywire. So we've been working on this, and in mice, what we did was we decided to remove the activator, smoothened. I mentioned this protein smoothened. So we take out the action side of this pathway in stromal cells. And when we did that, we created mice that had really 2 major features. One, they don't have a capsule. So I don't know if any of you wonder, like, what's a kidney like if it doesn't have a capsule? Now, I never thought about this. But what happens is the cells begin to form the capsule, the stromal cells, and then they don't proliferate. And so it's like a naked kidney. And when you do electron microscopy, the glomeruli are just sort of hanging out there. They're like me dancing, you know, like really, they're just hanging out. So if we could do hemodynamics on that kidney, it would be pretty interesting to see the way nephrons would be regulated. The other big feature is they have about half the number of nephrons. They're small, and they have about half the number of nephrons. So they don't have a capsule, and they are nephron deficient. So we began to, so this is be the typical way that we, in the modern era, go after this kind of problem. So what do? We take these kidneys and we analyze all the RNA transcripts that are being expressed in the mutants versus the controls. This is called RNA sequencing. When we did that, besides the ones we thought we would find would be deficient, like the one we knocked out, so that's a good sign, and the stromal cells themselves. But the one that was most affected was TGF-beta 2. TGF-beta is a member of the TGF-beta superfamily. And so we decided we need to test the function of this gene. What's it doing, actually? And so we decided to knock out its receptor, which is absolutely required for its signaling. And we did this in every lineage in the kidney that's important for development. So I showed you these lineages. The reason I did it is because we then, a graduate student in my lab made mice where TGF-beta receptor 2 is absent in the collecting system, in the nephrogenic cells, in the stromal cells, and in the combination of the stromal and the nephrogenic cells. And so we got a lot of information out of that, but to really sum it up, and this is like taking somebody's 3 years of work and putting it into a box, so I'm sure they'd be very appreciative of that, is we developed a model, and that is that in the stroma, hedgehog signaling regulates this receptor, which somehow talks to nephrogenic precursors, controlling the number that are formed. And so the next question was, what's it acting through to do that? And to do that, we then took these very complex mice that lack TGF-beta receptor 2 in these lineages. We again did bulk RNA sequencing, and we found a molecule that we'd never heard of. It's called cell adhesion molecule 1. Personally, I'd never heard of it. It's part of a group of cell adhesion molecules, or CAMs. We then did something called single cell RNA sequencing. So this is like you go in the grocery store, you take a box of cornflakes, you take the barcode, and you put it on the cash register, and they know it's not a bar of soap because it's got annotations in the barcode that tell you it's cornflakes, it's not soap. And so if you go through all the cells in the kidney, they all have barcodes. Now, I'm being simplistic, obviously. These barcodes were created by all kinds of information about which cells express which genes and therefore have which identities. But this is basically what we do. It's a very expensive experiment. And so we did this with mutant mice, and what did we learn? Well, we learned that actually one of the most regulated genes in the stromal cluster was this gene CADM1. So here it comes up again, which is satisfying. Then we hunted down all the genes in the genome that could be regulated by TGF-beta signaling, because they all have SMAD receptor sites. And it turns out there were only 3 genes that were changed in our data sets that were common to our data sets, and one of them was CADM1. So number 3 piece of evidence that helps. And then we looked by immunohistochemistry and we couldn't find CADM1. So, okay, this is getting to be pretty strong evidence that CADM1 is regulated by this pathway and is absent in these mice. So then we said, well, let's remove CADM1 because we don't know what it does in all these lineages. And so we did that. And there's another graduate student in my lab. So now this is 18 months. And 18 months later, a graduate student comes to me and shows me all the data. Well, we talked in between, I assure you. All the mice are normal. So you're like, well, is that good news or bad news? Now, for graduate degrees, that's very bad news. For science, that's good news. But science doesn't really like normal stuff. So this is a bit of a problem. But when we knocked out CADM1 in stromal nephrogenic precursors or in the combination, the mice are normal. But when we knock out TGF-beta receptor 2 and CADM1 in any of these lineages, trouble abounds. So when you knock out both, then you have defects in the cortex, in the stroma, but also the nephron number is depressed. So the sum total of this, and this is the kind of kidney we get when we knock out both, and you see this is a normal late gestation kidney, the cortex is filled with glomeruli. And look how rarefied this is. I mean, it's sort of, the early structures are there, but it's very anemic, as we say. There's not going to be many nephrons. So the end of this is, here's a contribution to a series of stories that are being told in the literature about, now we have a new pathway that controls nephron number via hedgehog signaling, TGF-beta signaling, CADM1, to make nephrons. And it turns out that we've learned that TGF-beta actually controls the amount of CADM1 that's expressed. Turns out that TGF-beta receptor 2 actually interacts with CADM1 when we do these kinds of experiments. And it also turns out that TGF-beta controls a whole bunch of other adhesion molecules that we're now trying to understand how it controls like a program of adhesion. So, which is absolutely required, because if nephrons are going to form, cells have to figure out where to go and how to associate with each other. And if they can't do that, they can't make these structures. So lots of work ongoing in this regard. But this is a typical sort of story, I would say, for how people in an experimental sense explore the idea of how do you make nephrons, what are the morphogenetic principles that underlie it. Now, this is all well and good, and this information will be applied to big genetic subsets to ask whether these genes are involved or are variant in human populations as we get to look at nephron number, those who have low nephron number versus not, and whether there's certain variants that co-segregate with that. But I think what's been sobering over all these years is that when we look at congenital anomalies of the kidney, we basically thought that most of these cases would be associated with a genetic cause. And it turns out it's not true at all. It's only about 20%. And so where's the rest? And so I think we now posit that a mixture of epigenetic, environmental insults or exposures are involved in creating this, but we don't really know anything about it. And this is a major challenge. And I would say for those of you interested in biology, epigenetics is a major area for exploration in terms of all disease, but certainly kidney disease. We do know from mice that certain things do go on. And so I'm now going to focus your attention for a few minutes on what we understand is essential in mice, and that is the activity of enhancers that control gene expression. So every gene has a promoter. It's got a set of sequences. The transcription factors bind to these promoters and activate the gene. But there are also regulatory areas, repressive areas, enhancing areas that are absolutely required for the activity of these genes. And we know that certain proteins, and they're called histones, they bind to genes, and certain moieties, particularly lysine, in these histones have to be activated or methylated, in order for the enhancers to have their activity. And one of these are marked by, so this is histone 3, lysine 4, it's methylated. And another one is in histone 3 is lysine 27, which has to be acetylated. And these are absolutely required for nephron formation in mice. So again, we go to a human disease. So here we focused on a human disease called Kabuki syndrome. Kabuki syndrome features developmental delay, some skeletal defects, cardiovascular defects, and kidney defects. And this is a big, complicated protein, over 5,000 amino acids. I'm going to draw your attention to two major domains in this domain diagram. One is called the set domain. This is a catalytic domain for monomethylation of lysine 4. And here's a coil domain, which is like a platform that allows a bunch of other proteins to associate with KMT2D in order to allow for the acetylation of lysine 27. And we know that mutations in this gene are responsible for Kabuki syndrome. So that's what we know. A postdoc in my lab did a meta-analysis of 737 published Kabuki patients. And what we learned was that 35% of them have CAHKUT. CAHKUT is congenital anomalies of the kidney and urinary tract. Beautiful name, CAHKUT. And 46% of the pathogenic variants actually reside in the coil domain. So that's interesting. And that patients who have mutations in that domain have a twofold higher risk of having Kakut. So Sarah, my postdoc, she went to AlphaFold. Now AlphaFold's quite famous. If you follow the Nobel laureates, AlphaFold got the Nobel Prize this year. So AlphaFold is a program basically using AI, artificial intelligence, that allows you to take domains, protein domains, model them, and ask whether variants cause a difference in these domains. So here's a wild type domain, coil domain from KMT2D. It's three-dimensional demonstration. Here's one with a variant that we have in one of our patients that I'm going to focus on now. And so one of our patients, here's the modeling. And if you just stare at this a while, you'll see that the gold and the blue don't really overlap perfectly the way they should in the coil region. So before you get sick, dizzy, and so on, I'll move on. What we decided to do was we said, because we don't really know, one, what does this gene do, really? And number two, what about that coil variant? So what we've done, we've taken blood cells, so stock blood cells made induced pluripotent stem cells. We changed those iPS cells into kidney organoids. And in those organoids, we removed the set domain. So we removed the catalytic domain. So this protein can function. So we'll see what that looks like. And then we also took urine from the patients. So I like doing this because I deal with children. And I don't like taking blood from children unnecessarily. They don't really enjoy it that much. And so we just take their urine, which they're happy to discard. And we make stem cells out of their urine, which is actually quite efficient to do. And from that, we make organoids. And so here's a patient that was used for this purpose. And then we make organoids. And here are the organoids. So these organoids are made by various protocols, which we don't have time to really get into. This is a way of looking at an early fetal kidney, basically. It's not a mature kidney, but it's a fetal kidney. So here's a 25 days after starting a culture. Here's an organoid. Here's some markers used to show that we have the right cells in the right place. And here's the one that's called coiled. So it's small, and it's basically just a bunch of cells. So it's not very good at making nephrons. And here's the one that has the set domain removed. It's not very happy either. It's just, it looks different, but basically the same results. You can do quantitative PCR, look at all kinds of markers, and demonstrate basically the same thing. All the differentiated cells are just not showing up. And then you can do methylation studies, which are pretty interesting that we've done, because interestingly, the cells, the organoids, well, in this case, it's intermediate mesoderm before you get to organoids. It's the one that has the variant in the coil domain that really has a problem with methylation, much more than the one that had the catalytic domain. These are heterozygotes, by the way. That's what happens in a human disease. These are dominant diseases. One allele affected, one allele normal. So a lot to be learned about the biology, and where we go from here with these models is that we can now look at gene expression, the expression of genes that we know control kidney development. We can look at how the regulatory regions are functioning within these genes to really get some further insight into the, quotes, epigenetic landscape in a human kidney tissue in control and disease. Okay, so that was the sort of basic science portion of linking what are we doing in preclinical work to try to link in a translational sense what we know scientifically to what happens in human beings. So now this is the last part I want to talk with you about is my concerns about this phase of human development. And that is the last phase. If we look at preterm neonates, we're going to see the numbers. So as I said, nephrogenesis ends at 36 weeks. 60% of nephrons are formed in the last trimester. So when does the last trimester start? 28 weeks, 27 weeks, So that phase, 27, 28 weeks to term, is like a really critical period of making nephrons. And let's be clear, to date, we cannot make new nephrons after term. Now, we can take, preterms will make nephrons after birth for some period of time, variable amounts of time. That's been demonstrated, but we can't engineer new nephrons. We don't know how to do that. So here's the curve of nephron formation. And there's a lot being formed here. And then there's epidemiologic evidence showing that pretermers are at risk, extreme pretermers, meaning they're less than 28 weeks, are at risk for chronic kidney disease. And it turns out that in a big national study here called the C-KID study, there's a lot of pretermers who've been enrolled, which is interesting because they have chronic kidney disease. It's not because they're just pretermers. Let me also emphasize that there's an interaction in the preterm area between normal weight and low birth weight. So we'll come back to that. Some preterm infants are lower weight than they should be given their gestational age. But here's prematurity data, which I think is a wake-up call. So here are the number of pretermers born in the United States. This is March of Dimes data, very good data, March of Dimes. So 2020, 364,000 preterm births. That's 10% of all pregnancies. Live births, 10% are preterm. Less than 28 weeks. So this is the critical period that I'm talking about. 55,500 per annum. Average survival rate, 70 to 80%. So I can tell you, because I go in the nursery all the time. In this world, a lot of these babies are doing really well. And we think they're fine. Well, we know they're not so fine, but they seem fine. But I don't think they're so fine because they have been born at a time when nephrogenesis is by far not complete. And even though they may have survived in other areas, I think they're at great risk. And if we look at this internationally, here's a study of 194 countries and regions from 2010 to 2020. 13.4 million babies born less than 37 weeks gestation, less than 28 weeks, 567,000 per annum. And if we look at Southern Asia, Sub-Saharan Africa, almost 65% of preterm births globally. And we need to adopt a global mindset around what's happening to these kids, not just a Canadian mindset or an American mindset. And given that our populations are mixing so much, we live in a world of migration and movement. Certainly in my practice in Toronto, we see a lot of kids from elsewhere every day. Now, preterm birth is linked to disease in later life. So now this is for you. Here's a Swedish study, 4 million people in a birth registry. Preterm survivors, increased risk of developing CKD. Extreme preterm, less than 28 weeks at highest risk, and the hazard ratio is shown. And the risk is not apparent. So us pediatricians who are looking after children and adolescents, we may not see any evidence of this unless we really look hard. We don't have great tools. But you, on the other hand, it will come because stuff happens to people. And I think the phenotypes will emerge. But we have lots of gaps. Sex differences. What are the risk factors? What are the mechanisms that control cessation of nephrogenesis? How do we measure nephron number? So one of our big challenges, we don't know how to measure it. We have no idea, actually, in a human being how many nephrons are there unless we do an autopsy. So it's a big problem. We have size and volume of kidneys, but these are rather poor correlates for nephron number. So that's a big problem. People are working on this, but if you are working with a bioengineer, this is a great project because we need ways to do this in vivo. And we need mechanisms to identify those at risk for chronic kidney disease, because not everybody's at risk. And why are some people at risk and not others? And in diabetes, I'm part of a big heterogeneity project in the United States to understand why are some people getting diabetic nephropathy and not others, for example. And then the mechanistic link between nephron number and CKD. These are huge issues. And then to load it on, just so you won't feel too good about this, I want to add the problem of low birth weight, because low birth weight is a compounding factor in some of these babies. And the issue, and I'm watching time, first of all, there's strong epidemiologic evidence that low birth weight is again associated with chronic kidney disease. And there are a number of factors that are associated with that, and including, here's meta-analysis of showing that with low birth weight, increased risk of type 2 diabetes in children, increased risk of cardiovascular disease, increased risk of hypertension. All these things are true and compound the problem. And so this is where we get to the Barker hypothesis. David Barker, who observed famine in Dutch populations and posited that the lower the birth weight, the higher the risk of death secondary to cardiovascular disease in later life. And why? Because it was hypothesized that in utero, in the face of starvation, that the baby undergoes metabolic changes in order to defend itself against deprivation. But these metabolic changes come to rear their ugly heads in the face of excess nutrition after birth. That's the fundamental basis of the Barker hypothesis, such that it leads to generational effects of metabolic abnormality, including diabetes. And so the Brenner hypothesis, which I showed you, which is low nephron, has been joined to the Barker hypothesis. So low nephrons, increased metabolic change, leading to difficulty with respect to kidney outcomes. Final piece is what happens in the nursery also has its effect. And we know that lots of things happen to babies after they're born. And the AWAKEN study is a good example in pediatrics of how this has been demonstrated. But being born 28 weeks or less in a nursery is not the same as being in utero. How much oxygen, what kind of nutrition, what kind of drugs, infection, and so on and so on and so on. These are also risk factors that need to be understood. So, basic thesis for this talk has been, well, we know about this, and that's enough trouble for us. There's lots of disease that's in our face, big problems, but there's a lot of this coming down the road that we're not really so aware of because we can't measure it, we don't see the phenotypes immediately, but the issue of low nephron number is a real risk for us as we're thinking ahead. And finally, I think I've already summarized that these early life insults producing low nephron number and metabolic derangement are serious risk factors for the occurrence of chronic kidney disease, which will unfold in the way that you're aware. Hemodynamic issues, loss of cells, and then loss of filtration capacity. And that's the good news for today. So, channeling Barbara Murphy, I thought, well, what are 4 things that you guys could work on for the rest of your lives? So one is maternal health. I mean, we have to be a lot more serious about maternal health globally, because this is clearly an underpinning to the whole situation. The occurrence of preterm birth being so high, 10%, is often due to poor maternal health status. So a lot to be done there. You don't have to be a nephrologist to worry about maternal health. I'm sure it'll help though. Figure out how to measure nephron number so we can get a lot more specific about what we're talking about. Elucidate mechanisms. You know, you don't have to know everything about mechanisms to know what to do, but it sure helps. And so I think we need to understand the natural variation in nephron number with respect to what progenitor cells do, how nephrons form, and how they stop being formed, the triggers for premature delivery, how fetuses really adapt, and the heterogeneity that could guide us towards precision and advancing equity across all populations. And finally, make kidneys. beyond their natural potential, that would be nice too. And organoids are not there. So organoids are a nice start, and they've taught us a lot of science, but we've got a long way to go. So if you choose any of these things, I think you'll be very well occupied for at least 30 years. And with that, I want to thank, there are members of my lab here cited in red who've been part of the stories that I told you. But more importantly, I want to thank you, and happy to take your questions and comments. And for those of you online, this is what you do to tell people you were there. Thank you.